Well, hey guys, welcome to the review for test one. We're going to try to go through the review as I have planned it. This is the way I teach it for my in-person class. And if you feel like you're missing something or there's some gaps, let me know. I can go back and offer some mini lessons on what we missed. Sorry I'm late getting this to you. I've been battling a cold this week. Thankfully, I'm feeling better now. But anyway, let's get started. So the way test one is going to work is the way all of your tests are going to work. Whereas your quizzes are built into my mastering biology, and they're mostly multiple choice or working through the modules. These tests will be hosted through Blackboard. So it'll be a test through Blackboard, and they'll be short answer style questions. I want you to use your notes. You're going to have an hour to take, hour 15 minutes to take the test, and I want you to be able to use your notes. Obviously, you guys are at home. You've got more resources at your discretion. I'm not going to use the on and lock system. I'm going to trust you guys. But the tests are going to, or test questions are going to be much more critical thinking. So I want you to kind of explain your reasoning, see some personal examples there, and kind of work through the module. Like I said, you're welcome to use your notes, anything from your studying that's helped you. You can even look at your textbook. But the goal of these is to help you think about the material rather than just memorizing facts. I want you to be able to think critically. Remember, that's one of the big goals of this course is that you engage with biology and the sciences in general with a critical thinking lens. Most of you aren't going on to be a science type major. You're going on to be an educator, a psychologist, a business owner, a criminal justice expert, right? A number of things. But all of you are going to be living in this world and making decisions based on your worldview and based on your understanding of science, right? So my goal in this course is to help you understand science a little better, particularly biology. Think about your worldview more intentionally and think about how worldviews kind of mesh up with science, particularly peeling apart naturalism from science, like we've talked about. And then be able to go and fact check. So when you got to make decisions on your healthcare, you got to decide whether to pursue that cancer treatment or whether to take that vaccine or any number of decisions. You're going to lean on your worldview. You're going to lean on friends that you count, but hopefully you're going to lean on your understanding of biology and your ability to find credible sources. So that's the biggest goal of this course. So let's talk about test one. Test one is going to have 4 questions with a bonus kind of tacked onto the fourth. Again, these are going to be multi-part questions, so it might be 4A, 4B, 4C. But the way these questions work, they are a combination of essay style. Particularly the first 2 questions will be essay style, where you're kind of synthesizing some of what we talked about in our first discussion and what you talked about as you read the Bloom text and did that book reflection. Then the other 2 are going to be working through a scenario where you practice first your understanding of the scientific method and be able to kind of think through that and do science well, at least kind of theoretically or hypothetically on paper. And then the fourth one's a big one. This is going to tie in the chemistry piece. Now, I know that was a big chapter or a big unit rather. Most of you guys did well. Don't panic. In the grand scheme of things, chemistry is a minor role in this course, but you can't really understand biology well without having a basic understanding of chemistry. So this fourth question is going to get at your understanding of some chemical properties, what the difference in hydrophilic versus hydrophobic, different types of bonds, chemical bonds that bind elements together into compounds, and then thinking through the cell. So one of the things you learn in this week is the major organelles of the cell, basic cell theory, and then kind of how these things work together. So anyway, I'm gonna try to lecture a little bit in this. So this will be a little bit longer than a typical review, but hopefully that's helpful. So the test will be like that. Question 1 is going to be about defining science and explaining its purpose and limitations. Question 2 is going to be about describing the biblical concept of imago Dei, or being made in the image of God, and kind of thinking about how that impacts how we do science, more particularly how that impacts worldview implications, right? The goal of that question is helping you think about the fact that science only gets you so far. Then you're going to have to lean into your worldview to make decisions when it comes to kind of applying science. Third question is going to be working through the experimental process. So I'm going to give you a basic scenario, ask you to write a hypothesis about that, design an experiment where you would test that, and then analyze data where you would draw conclusions. So I'll have you actually sketch up some graphs and analyze some data from graphs. And then the last question will be thinking about a scenario that talks about proteins in particular, and using that as a model to kind of think through the what we call the endomembrane system of the cell. Basically, what that means is how particles, particularly in this case proteins and forming proteins, move through the cell through that membrane system, starting in the nucleus with the gene that codes for that protein, to the RNA that's a copy of that, to eventually the final protein that will be secreted from the cell. All right, so here we go. Question one, what is science? What I'm looking for here, and again, the question will be worded a little bit differently, but what I'm looking for here is your ability to kind of define science well, understand the purpose of science, the limitations of science, and then to kind of peel apart science as a limited tool of understanding about the natural world, and then naturalism as this kind of comprehensive worldview that leans on science. I hope that makes sense. Those aren't one and the same. Textbooks and kind of mainstream talking about science, we often conflate the two, but fundamentally they're not the same, right? Science is a tool, and naturalism is a worldview, right? Science is a process of understanding about the natural world. It tries to explain why and how natural phenomena occur. It's through this process of deductive reasoning, through the scientific method. But science by itself is a tool, right? It only gives us evidence that then has to be interpreted and filtered through a worldview, right? So that's where naturalism comes in. Now, naturalism is a very science-dependent worldview, right? Naturalism is this worldview that rejects the idea of supernatural. It's a worldview that assumes everything that exists can be explained through nature. Everything that exists is natural. There's no guided mechanism. There's no creator. Nature is all there is, and nature is all there will always be, right? That's kind of the fundamental belief system of naturalism. So then that's different than science, right? Science itself just says, hey, how does this natural process work? Naturalism goes a step further and says there can only be natural explanations. There is no supernatural. Nature is all there is. I hope that makes sense. So again, naturalism is a very science-dependent worldview, but science by itself doesn't have to be viewed through naturalistic lenses, right? Christians, atheists, Buddhists, you name it, can look through science, look at science, and then filter it through their perspective worldview. So be able to do that, be able to kind of give a definition of science, and then distinguish that from naturalism. Before we go on to the Imago Dei question, one thing I wanted to add about the understanding the science process is also the limitations, right? So science isn't just limited by those worldview implications, right? In other words, science has to be attached to a worldview to give a comprehensive view of reality. That is a limitation, right? So kind of saying that another way, science itself can't answer ethical, metaphysical, spiritual questions, right? That's just not the way science works. Science is a kind of a measurable process of understanding natural phenomena, and it's limited to that. But science is also limited by bias. We talked about this idea that everything that we do in science, the questions we ask, the money that funds the research, the way we interpret the data, all that's going to be affected in some way by our bias. Now, ideally, we mitigate that through peer review, through kind of assuming professional thinking. But humans are humans, right? Science is done by humans. So as much as we try to check our bias and disclose our bias, we never eliminate our bias. So keep that in mind. Bias will always affect science to a certain extent. But then there are others, right? Science, when we're doing an experiment, for example, we can only control variables that we know are variables, right? Some variables we don't know are variables, so we can't control them. And they might be confounding variables, right? They might be variables that actually affect our data and cause us to make faulty interpretations and conclusions. But there's some things that even if we know they're variables, we can't control them, right? An example would be meteorology. As much as we can model storms in a lab, we can't fully recreate the Earth's atmosphere and all the factors that go into driving storms, right? The best we have is models. We can take what we know about certain factors and historical trends about how storms behaved kind of in the real world, and we can make models that guess, that project, but we're never going to be certain, right? Because we simply can't control those variables. So there are some variables we just can't control, and then others, right? Like sometimes when we isolate variables from the real uh, natural phenomena, right, the real world, they behave a certain way in the lab, but when those variables are then back in nature, the phenomena would be different. So anyway, just think through some of those limitations and be able to articulate some of those limitations of science. Imago Dei is a Latin term that means in the image of God. We talked about this in our previous lesson. I want you to just have a good understanding of this, that as Christians, as people who are trying to think through a biblical worldview, Scripture makes it pretty clear that God created all things and they were good. But then God created humans, man and woman, and they were very good, right? And he specifically said he created us in his image. We talked about some of the things that meant. I gave you a John Piper quote. But the idea here is that we are physical and spiritual beings, and because we are God's image bearers, we are made to bear his image, right? We have responsibility to God. We can sin against God, and Adam and Eve did, and so do all of their descendants. And so that kind of sets up all of the biblical narrative and our understanding of the gospel. But I want to hone in on the idea of being made in God's image and just think about some of the practical ways that impacts how we as Christian scientists, people who attach science to a biblical worldview, would think differently than someone who has a different worldview, right? Uh, we we think differently about human life, for example, right? Regardless of your opinion on abortion, I know that can be a complicated issue on both sides of the argument. But at the end of the day, abortion is taking a human life, right? And so, as from a biblical worldview, we would view abortion as um, taking a human life, and that carries more weight than taking, say, the life of a bacterium, right? If you're treating an infection. Stated another way, we believe that humans are babies, particularly, are not parasites, right? They are humans that God has designed to be made uh, through that marriage, through that union, through that fertilization, and He's working miraculously to cultivate a new being and a spirit inside the mother's womb. Anyway, that's just an example. So all that to say, think about the reality that if we're made in God's image, that has implications. It also has implications on how we think about nature, right? We are created from this earth for this earth, and so although heaven is our home, the picture that we get, and I think I told you guys in the previous lecture, is that in Scripture that God is making a new heaven, a new earth, right? One day He's going to bring heaven and earth back together. There'll be no more sin, no more death. We'll live in resurrected, perfected bodies and reign with Him for eternity and enjoy Him forever. The creation mandate, right? God created Adam and Eve and told them to be fruitful and multiply, to fill the earth and subdue it. The idea there is stewardship, right? And so uh, thinking about humans being made in God's image has stewardship implications as well. We have some level of responsibility to manage creation well, right, as God's image bearers. It also has implications for how we think about science in terms of being creative, making good discoveries, right, using the talents that God's given us well to bring Him glory and to serve others well. I think there's a real expectation there that we would use science and use even modern medicine in a way that honors God and helps the human condition. I think that is a implication of Imago Dei. Okay, so then doing science well. Remember this process, right? Scientific method is based on observations. We use those observations and check the literature, right? We do some searching to find previous understandings through textbooks, through peer-reviewed articles, and that's going to lead us to form questions. We're going to check those questions to see how other people have answered them. Eventually, we're going to form a hypothesis, right? So our hypothesis is our tentative answer to our research question. So on the test, I'm going to give you essentially a research question and ask you to write a hypothesis about it and design an experiment and give me some example data, right? So you should be able to do that. When you take that question and translate it to a hypothesis, your hypothesis is your predicted answer. So it's directional. You're predicting that there will be a change or there will be no change or something like that. And it's a statement, right? It's no longer a question, it's a statement. You're stating your predicted answer. Then you're going to test it. So remember the elements of a good experiment. We've got to take our sample, make sure it's a representative sample, ideally break it into two groups randomly assigned. We also want to make sure that sample is large. So when you're thinking about the test question, make sure that there's enough participants that your data are meaningful, right? If you only got 5 or 10 participants in a clinical trial for a drug, it's probably not going to be that conclusive, right? You can't generalize your findings about these 10 participants to the entire 8 million or 8 billion world population. Instead, if you had, let's say, 100 participants or better, 1,000 participants, right, and you randomly assign them to groups, then you're going to have a little bit more confidence in your findings because you've got a larger, more representative sample. Anyway, we're going to measure our data. We're going to try to control any other variable, right? We can do that through randomization, but we can also do that through only manipulating one variable at a time, right? Keep everything else constant. So if we're doing a clinical trial for a drug, we're going to have a control group that gets a placebo, right, essentially a sugar pill, and an experimental group that would get the drug. And then in your test, think through that process, right? How long would they get the drug? How long would the study go? What exactly would you measure to see if the drug's working? That's what I'd be looking for. And then I ask you to kind of draw some example data based on what you said you'd measure, and then give you some other data that might look at side effects or other things you might measure, and just being able to interpret those graphs well. So again, here's a summary of that. In this example, we've got a new blood pressure medication. We want to see if it affects humans well, if it actually lowers blood pressure. We found that it works in rats as an animal model, but we want to see if it actually works in humans. So we're kind of starting the first stage of a clinical trial. We're going to take a sample, a fairly large sample, and then we're going to randomly assign half of them to the experimental group that would get the drug, and half of them to the control group that would get the placebo. They're not going to know whether they're getting the real drug or not. Ideally, the researchers wouldn't know whether they're who's who, so there's no bias there and placebo effect. And then we're going to, whoops, let me just skip slides there. We're going to implement the study, right? In that case, they would take the drug, let's say every day. We'd measure their blood pressure once a week, and at the end of the trial, we'd take their average blood pressure, and we'd look at the average blood pressure for the control group, the placebo, and the average blood pressure for the experimental group, the drug, and we try to see if one was lower than the other, and if it was statistically significant. And if it was, we would conclude that the drug worked. Again, in the clinical trial, you're also going to monitor side effects, reported headaches, reported cases of nausea, reported liver damage, you name it. But anyway, that's the idea of the scientific method, right? So we use our data to either support or reject our hypothesis, and eventually that could form a theory, right? Or at least help us apply that theory or reshape that theory. All right, here's the last one, and this is a big one. Last week and this week, we talked about, or you learned about in your reading and your homework, basic chemistry. So all of the elements on the periodic table, those are the elements that exist in nature. We talked about subatomic particles, elements, or electrons, protons, and neutrons. There won't be a lot of that on the test, so don't panic too much. Although I want you to have a basic understanding, right? That the neutrons and the protons are the ones that give them mass, and they're in the nucleus. And then the electrons and protons are the ones that kind of determine the charge. And most atoms, when they're in their elemental form, their natural form, they are non-ionic, meaning they're the same number of electrons as protons, and so they are balanced out. Remember, electrons have a -1 charge, protons have a +1 charge. But then if an atom gains or loses an electron, we call it an ion, right? And now it's charged, right? Either negatively or positively, and that's going to give it properties, right? It's going to make it water soluble because it's going to make it polar. It's going to make it able to participate in polar bonding, ionic bonding. It's going to give it electrical properties in some cases, right? And be an electrolyte. So that was kind of the basics of that. Then we talked about biological molecules. So we kind of built onto that this idea that atoms, by interacting with their electrons, particularly the valence electrons, the ones in the outer shell, they can form compounds, right? Form molecules that are made from other elements. And that's the building block of chemistry, of life, right? So we get these larger molecules, and then when we think about molecules that are made by living organisms, those organic macromolecules, we get things like proteins, carbohydrates, lipids, nucleic acids, right? And those are the building blocks of life, right? Those are unique to living organisms, and those are the kinds of molecules we'll be talking about in biology. So go back and make sure you have a basic understanding of proteins versus carbohydrates versus lipids. In general, proteins are the workers of the cell, right? They are functional molecules. They're going to be signal molecules. They're going to be enzymes. They're going to be transport proteins in the membrane, etc. Fats in general are going to be long-term energy storage, but they can also be used for hormones. They can also be used for the cell membrane, right? Carbohydrates in general are short-term energy molecules, right? Particularly glucose. Most living organisms are primed to use glucose metabolically at the cellular level. But carbohydrates can also be used, by being attached to proteins to give them certain characteristics. And then nucleic acids, aka DNA and RNA, those are the information storage molecules primarily. Although some nucleotides, like ATP, for example, can be used to carry energy. So go back and look at that. Make sure you have a good handle. But on the test, we're going to focus on proteins. One of the things that you should have caught in your reading that we'll see kind of come up again and again is that when it comes to molecules, particularly proteins, their shape determines their function, right? And ultimately, their shape is determined by the amino acid sequence, right? So proteins are made of amino acids. There are 20 amino acids that are used to make proteins in living organisms. Think about that analogous to letters in a word, right? In an English alphabet. There are 26 letters in the English alphabet. And just like arranging those letters in words and then those words into sentences gives meaning, right? Similarly, the way we stick those amino acids together in a chain of amino acids will give that protein a particular shape and function, right? I hope that makes sense. And all that has to do with three-dimensional nature, right? So these proteins will be made by attaching all these amino acids together in the cell, forming that polypeptide chain, right? We're going to make covalent bonds between all of those amino backbones. And then as that protein chain is emerging from the ribosome in the cell, it's going to fold up, right? And most of the cell contents is water, right? At least the background, the cytoplasm is water. So when those proteins, those amino acids encounter the water, some are going to be hydrophobic, meaning they're going to kind of be repelled by the water and fold in. Others are going to be hydrophilic, meaning they are polar, so they mix well with water, and they're going to be outward facing. Some are negatively charged, some are positively charged, some can participate in hydrogen bonding. You get the idea. And as all those amino acids come together and encounter water, they're going to fold up into the functional protein. So this question here, we're going to think about some of that. So think about proteins, but we're also going to think about this idea of cell organelles and even the flow of information. So this is a big one, and we'll see this theme throughout. So let's talk about this first question. This is just a kind of a review question. The question on the test will be more short answer, comprehensive. But let's read this. Erythropoietin is a protein hormone that stimulates red blood cell production. Okay, so that I'm just giving you information there. This particular protein is called erythropoietin, and it serves as a hormone, but it's a protein, right? So it's made of amino acids. It's going to have a particular shape. And its job, once it's released into the bloodstream, is to go to stem cells and red bone marrow and make red blood cells, right? That's its job. So if your body needs more oxygen carrying capacity, you're low on red blood cells, or you're in a new climate where there's less oxygen density, your body's going to make this hormone to stimulate red blood cell production. Anyway, EPO normally has monosaccharides attached to it. What is the correct order of structures that EPO will pass through? So Monosaccharides, those are the building blocks of carbohydrates, remember. Uh, and monosaccharides might be added to proteins, right? So protein modification happens in the Golgi apparatus. So in this example, you kind of work through, and the right answer would be we're going to start in the rough ER, right? So once the gene that's in the nucleus, right? The gene is that sequence of DNA that encodes for that particular protein. Once it's been copied into an mRNA molecule, it's going to leave the nucleus and go to the endoplasmic reticulum, particularly the rough endoplasmic reticulum. That's where the ribosomes are. That ribosome is going to translate that mRNA into the corresponding amino acid sequence protein. And then it's going to be packaged into a vesicle, right? This little membrane-bound truck that will carry things inside the cell. Then it's going to go next to the Golgi apparatus. So the Golgi apparatus is distal. It's farther from the nucleus, kind of headed towards the cell membrane. And the Golgi is where proteins are modified. We might add monosaccharides. We might add some other chemical residue that might give it function. That might tell it where it needs to go in the cell. But if that happens, it happens in the Golgi apparatus. And then finally, it'll be packaged into another vesicle, and in this case, sent to the cell membrane to be released from the cell. Okay, we'll come back to that in a second. So remember, here's our breakdown of biological molecules. All biological molecules are organic, meaning they are made by living organisms, right? Hence the name organic, and they're based on carbon. And those biological macromolecules that we think about are carbohydrates, proteins, lipids, and nucleic acids. So we talked about this too. Proteins ultimately are a string of amino acids, right, that are hooked together by covalent bonds. When the amino group and the carboxylic acid group of two amino acids are linked together, and then we've got those functional groups kind of hanging out, facing the outside, and that's what's ultimately going to fold together. So follow with me here. This first picture is our primary structure. This is our primary structure. So this is the amino acid sequence, right? So as the protein is being made, it's just the order of amino acids, much like the order of letters in a word. This is the order of amino acids in the protein, but that's going to start folding, right? So as those side chains, those functional groups on those amino acids start interacting, some are going to hydrogen bond, some are going to be hydrophilic, hydrophobic, so we're going to start to get folding. This second one here, we kind of have two motifs that proteins can fold into, this coiled structure or this kind of folded structure. You don't have to memorize that, but that happens first. But then ultimately, when we get to here, we get a fully folded protein subunit, right? So that protein is fully folded and it's functional. Now, some proteins, like this one, this is hemoglobin, are multi-subunit proteins, meaning there are multiple amino acid chains that will be folded and stuck together. So you can see that here. That would be the quaternary structure, where that fully assembled protein is folded and it's ready to go. But the big thing you want to remember here is that structure determines function, right? And so if we have a mutation that changes one of the amino acids and it ends up encoding for an amino acid that's different, right? Let's say it was an amino acid that's positively charged and now it encodes for an amino acid that's negatively charged, that's probably going to make it fold up differently and therefore be non-functional, or at least alter the function, right? So structure determines function. If we change the shape of that protein by giving it a different amino acid that kind of puts a kink in it, we're going to change the function. All right, so this is the last one here. I know this is a lot. Hopefully this is helpful. I'm trying to rapid fire go through the big things. So question 4, we'll kind of get at some of that basic understanding of proteins, but it's also going to get at your understanding of the cell. So hopefully you've done the homework, you've read it, you've got a good understanding of the cell. But let's quickly go through the cell with a metaphor that I think will help you make sense of this. Think of cells as this complex ecosystem. It's this complex city, and it's a manufacturing city, right? It's a manufacturing culture. On a side note, cells are the simplest unit of life, right? All living things are made of cells. Some are single cell, like bacteria, protozoa, but many are multicellular, like plants, animals, fungi, and even some protists. Regardless, in a eukaryotic cell, and again, that's most multicellular organisms, or all multicellular organisms are eukaryotic, that's plants, animals, fungi, protists, they're going to have these membrane-bound organelles. But we also have simpler cells, right? We've got prokaryotic cells, bacteria, that are smaller. They can't support multicellular organisms, body shapes, but they have all the basic components of a cell. They've got a cell membrane, they've got DNA with the genome, they've got ribosomes and the ability to make proteins, right? But they're just simpler. Most of the cells that we'll talk about in this class, and this model here, follows eukaryotic cells, because most living organisms are eukaryotic. Eukaryotic cells are more complex. They've got membrane-bound organelles. Organelles are these functional units inside the cell, again, inside this city. Okay, so that's the basics of a cell. Let's think about this metaphor. There are a few organelles that I want you to know. Obviously, the main one is the plasma membrane or the cell membrane. All cells have those, and that contains the cell, right? It forms this semi-permeable barrier between the outside environment and the internal environment, right? It creates the capacity for the cell to be autonomous, to be self-contained, right? To have a different environment inside the cell than outside the cell. And because it's made of phospholipids, right? They are these special lipids that have these lipid tails that are hydrophobic, right, like you would expect. But then they kind of face together like this, so we make this lipid bilayer. But then the outside and the inside of the cell membrane, where those phosphate heads are, those phosphate heads are not lipids, right? It's a phosphate molecule, it's an ion. And you know, ions are polar, they are hydrophobic or hydrophilic, they are water loving. So these phospholipids are unique in that they are what we call amphipathic or bipolar. They have two natures, right? One side is a lipid tail, and it's hydrophobic. It's going to hide from water. And then the other side has that phosphate group on it, that phosphate head, and it's hydrophilic. It's going to interact with water. And so when those form the layer like that, we kind of get this lipid bilayer, right, with the phosphate facing in and out. And so that makes the cell membrane relatively impermeable, right? Most molecules are too large or polar, and they can't just diffuse across, right? So that gives us the capacity to control what comes in and out. And of course, proteins get involved, and we can use that. Anyway, let's back to this question. So I want you to be ready for the test question. The other organelles I want you to know, aside from the plasma membrane, are the nucleus, the endoplasmic reticulum, the Golgi apparatus, of course the mitochondria, but they won't be in this particular example, and then vesicles. So think back to that metaphor I gave you of a manufacturing process. Let's say you wanted to order, I don't know, a custom pair of tennis shoes from Nike, and you bought them on Amazon, right? You're going to have to put in your order. They're going to send that order to the engineering department, right, where the blueprint of those shoes are housed. And then we're going to copy that blueprint, right, send it to the manufacturer, make that shoe. Then that shoe is going to have to be packaged and sent to wherever it's customized. Let's say it's customized in-house, but that's another floor, right? To be customized to your specification. And then it's going to be packaged and sent to the warehouse, the distribution center, or the Amazon distribution center. And it's going to have to be tagged with your shipping label. It's going to have to be inspected, make sure it's good to go. And then it's going to be put on a truck and delivered to your house, right? Let's think about that as a metaphor for how proteins are made in the cell. All right, so here we go. This is the nucleus right here. Right? This little spot here is called the nucleolus. That's just where ribosomal subunits are made. Remember, ribosomes are those little enzymes made of RNA that make proteins. Anyway, all this stuff in here, this is DNA, right? So it all starts with the gene. Think about the nucleus as the engineering department. And the genes, right, the DNA, are the blueprints. So the cell is going to get an order, and this will be a signal, right? But let's say this cell has gotten a signal and says, hey, we need to make this protein. Let's call it insulin, right? This is a cell in your pancreas, and you need insulin. You need to make more insulin so we can get your blood sugar down. So that signal goes to here and says, okay, make a copy of the insulin gene, right? So that's the first step. The nucleus is the engineering department. That's where it's going to happen. We're going to make a copy of the gene. We'll talk about what that is later. That's going to be a molecule called mRNA. But that copy of the gene will be made and sent out of the nucleus to the Golgi. That's what this is right here, this blue thing wrapped around the nucleus. Or excuse me, the endoplasmic reticulum, I'm sorry. The endoplasmic reticulum, here, I'll abbreviate that here, ER. And you can see the flow chart I have here on the bottom of the screen. Right? So next is the endoplasmic reticulum. That's where we're going to take that mRNA copy, and it's going to encounter a ribosome, right? So this will be the rough ER. And that ribosome is going to translate that message into the protein. It's a pretty fascinating process. We'll talk about it later, but that happens there, particularly if it's a membrane-bound protein or a membrane-packaged protein. Next, we're going to pinch off a part of the membrane and form what we call a vesicle. So you can see that here, this little sphere, this little membrane-bound truck. And that's the transport truck that's going to take the protein, or back to our analogy, the shoe, to the distribution center. Right. So this green membrane-bound organelle here is called the Golgi. And so, right, step 3 is we're going to package it into a vesicle and send it to the Golgi. Step 4, we're going to go to the Golgi, so that membrane merges with the Golgi, and now those contents are inside the Golgi. And that's where it's going to be processed for shipping, right? So back to our metaphor with the shoe, you got to box it up, make sure it's packed right. We got to put your shipping label on it, make sure address is appropriate, make sure postage is paid, and then ship it to your house. In an analogous way, proteins will be kind of put in their final form here. They'll even be tagged to where they need to go, whether that's in the membrane or outside the cell or some other part of the cell. And then once it's ready to go, we're going to package into another vesicle. These vesicles, like I said, are just little spheres that pinch off from the membrane. It's very dynamic, very fluid. So that's step 4. And we call it a secretory vessel because it's going to be secreted from the cell. It's the process of exocytosis. So then that last step would be that final insulin is going to be ready to go, packaged in that vesicle, and released through the cell, or from the cell, through exocytosis. Right. So then insulin's in your bloodstream. Your blood's going to carry it to your body cells that need it. Insulin's going to bind to insulin receptors, and it's going to do what it does, right? Get glucose in. Okay, that was a lot. Here's another picture of that, so you can see that endomembrane process. I hope that's helpful. Make sure that you spend some time studying. Go back and look at the structure of the plasma membrane like I talked about. Think a little bit about active and passive transport. Really briefly, because we've got this phospholipid bilayer here, right? Particularly this lipid bilayer here, right? So that's where the lipid tails are of these phospholipids, right? A single phospholipid looks like this, right? This part of the phospholipid is polar, meaning it's hydrophilic. It's going to love water, right? And it's going to interact with water. And then this part are 2 lipid tails, 2 fatty acid tails, and they are nonpolar, right? Meaning they're hydrophobic. They're not going to interact with water. And so then these are arranged in such a way where we've got a bilayer, right? So 2 opposing phospholipids are facing like this, and we've got this kind of double waterproof layer, right? These fats would essentially be waterproof. So if we look at that in the membrane, not many things can just diffuse across. They're going to be stopped, right? Water can't just diffuse across the membrane because it's polar. It's going to be repelled by these fats. Same with small ions. The only things that can just diffuse through simple diffusion across the membrane are gases, right, like oxygen and carbon dioxide, and then cholesterol-based hormones, steroid hormones like testosterone, estrogens, because those are small and they're lipid soluble, they can diffuse across. Everything else needs a protein, right? So that gets us to passive versus active. So simple diffusion just across the membrane, right? So a gas just diffusing is a form of passive transport. But so would be water through a water channel, right? So we've got these proteins that essentially punch holes in the membrane, and they're kind of like security checkpoints. Those amino acids that are in the lumen, the hole, the opening, are specific in chemical structure so that only certain molecules can get past them, right? Other things would be repelled. Kind of like a security checkpoint. So if you've ever been on a security base or a gated community, you don't have to pay the security guard, right? You've got traffic backed up behind you. You're going down the concentration gradient, so to speak. You're going from high traffic to low traffic. You're trying to get to work or get home. And you just need the right ID, right? As long as you have the right ID, you can get on base or get into your community. That's an analogy for facilitated diffusion, right? That's what this would be called, facilitated diffusion. It's a form of passive transport. And that's what insulin does, right? So insulin transports glucose. Glucose is a fairly big molecule, right? It's C6H12O6, and it is polar, right? So it's not going to get across the membrane. It's going to need a protein, and so insulin causes cells to make more glucose transport proteins, and then they can come across the membrane that way. Active transport is similar in that we need a protein, but it's different in two ways. One, we need energy, right? In the case of passive transport, the energy comes from diffusion, right? We've got a high concentration of molecules on one side, a low concentration on the other, and so they're just going to diffuse down their concentration gradient, right, from high to low. That happens in nature. Active transport needs ATP, right? So we're going to have to spend that cellular energy to cause this protein to work, right? Usually these are pumps. They're pumping things across the membrane against their concentration gradient, and it requires energy. So that's the second thing that's different, is that active transport, we can actually move particles from low to high, right? Kind of working against overcoming that force of diffusion. Okay. And then there's exocytosis and endocytosis. Again, that's membrane mediated. I hope that's helpful. Sorry this was so long. If you have any questions, please reach out. But good luck with the discussion this week, and good luck on the test starting Friday. Have a great night.